The topic that we should be dealing today is about the structure and the function of the immune system. It is very important for us to know what are the components that are mostly involved in the development of an immunity against different types of pathogenic microorganisms. So in order to understand the immune response towards this microorganism, it is very important for us to know what are the things that are involved. That is, what is the structure of the immune system and what are the different components of the immune system or the cells of the immune system that participates to eliminate the pathogenic group of microorganism to prevent the human from different types of diseases. So let us see what is the structure of an immune system. So before that, let us discuss what is lymphoreticular system. Lymphoreticular system is nothing but it is a system comprising of cells and the organs of the immune system. They are distinct group of cells, diverse group of cells that are involved in the production of an immune system or in the immunogenicity, and these cells are distributed widely in the different types of the lymphoid organs or the organs which usually develops this lymphocyte to produce an immune response or to mount an immune response against the pathogenic group of microorganism. So this lymphoreticular system usually consists of two important components. One is the lymphoid cell and the other one is called as the reticular endothelial component. Endothelial component. The lymphoid cells are the cells that are usually the one that belongs to the lymphoid cells are the lymphocytes and the plasma cells, which are usually the one that belongs to the specific immunity. That is nothing but the adaptive immunity or the acquired immunity. Now coming to the reticular endothelial system, which usually consists of the phagocytic cells. As we all know, the phagocytes are the cells that are able to ingest the pathogenic group of microorganism and cleave them into different fragments and inactivate the antigen. So phagocytes are the one that belongs to innate immunity or non-specific immune mechanism, whereas the lymphocytes and the plasma cells that belongs to lymphoid system are the one that mounts an specific immune response, whereas the phagocytes are the one that mounts an innate or non-specific immune system. And here the main important function of the phagocyte is to eliminate the organism by phagocytosis. Apart from being a component of the innate immune system, it also helps to activate the specific immune system by acting as an antigen presenting cell. So the lymphoreticular system usually consists of cells and the organs, among which we'll be studying about the cells, that is the lymphoid cells and the reticuloendothelial cells that are mostly used to mount an immune response. And apart from that, whenever there is an antigenic challenge or during an antigenic stimulus, our body or the immune system develops an immune response, and this immune response can be classified into two major components. One is called as the humoral immune response, the other one is called as the cell mediated immune response. So the humoral response usually involves the antibodies. The term humoral comes from the Greek word that is called as body fluid. So these antibodies usually are the one that circulates in the body fluid such as the blood and the plasma, and are able to mount an immune response towards the pathogenic group of microorganism. Because these antibodies circulate in the body fluids, they are called as or they are said to mediate humoral immunity, whereas the cell mediated immunity are usually mounted by about sensitized lymphocyte, mostly the T lymphocyte. So the cell mediated immunity is the one that can be mediated by the T lymphocyte, whereas the humoral immune response is the one that is usually mediated by the antibodies. Irrespective of whether it is an humoral response or the cell mediated response, both of them usually interact to produce a heightened immune response towards a pathogenic group of organism, and both interact equally, and they usually are the one that help to activate each other to develop or mount an immune response. Though they have different properties, but they interact to mount an immune response. So that is what is called as humoral immune response and cell mediated immune response. So the lymphoid system consists of lymphoid cells and the lymphoid organs. So first we will be dealing about the lymphoid organ, that is the organs of the immune system, where the lymphocytes are usually produced, they proliferate and differentiate to become immunocompetent to mount an immune response. So it is a place where the lymphocytes of different different types are usually generated. So here the lymphoid organs, based upon their role, based upon their functions, they can be widely classified into two distinct types. One is called as the central or the primary lymphoid organs, and next one is called as the peripheral or the secondary lymphoid organs. So the main role of the central or the primary immune response is to produce the lymphocytes, and they help the lymphocyte to proliferate and differentiate and get matured, and then these mature lymphocytes move on to the peripheral lymphoid organs to carry out their immune responses towards the different types of microorganisms. So the generation of lymphocytes usually takes place in the central or the primary lymphoid organs, whereas in the peripheral or the secondary lymphoid organ, they usually carry out their functions. So among the primary lymphoid organs or the central lymphoid organs, the examples are bone marrow, thymus, and bursa fabriceus. Bursa fabriceus and bone marrow are equivalent. So bursa fabriceus usually seen in the birds. That was discovered to mount an humoral immune response. A similar type of function was seen in the bone marrow of the human being. So it is the central lymphoid organs, which are mainly the thymus, the bone marrow, and the bursa fabriceus in the birds. So here the lymphocytes usually proliferate, they develop, they get mature to become immunocompetent to move on to the peripheral lymphoid organ. And these lymphocytes that have matured in the primary lymphoid organ move via the blood or the lymph in the lymphatic system to move to the peripheral or the secondary lymphoid organ. And here in the secondary lymphoid organ, during an antigenic challenge, the lymphocyte usually interacts with the antigen and mount an immune response. Examples of the peripheral lymphoid organs are the spleen, lymph nodes, and MALT, that is mucosal associated lymphoid tissue. So anything associated with the mucus layer, any organ that is associated with the mucus layer usually are under the mucosal associated lymphoid tissue. So there are variety of mucosal associated lymphoid tissue where you can see the lymphocyte mounting an immune response towards a pathogenic group of microorganisms. So broadly classifying lymphoid organs into two major types. One where the lymphocyte usually generate, they get produced, they proliferate, they differentiate, they get mature to become immunocompetent to mount an immune response, and this function is usually seen in the peripheral lymphoid organs. Now the first lymphoid organ that we'll be studying is about the thymus. Thymus is a flat bilobed organ that is usually present in the upper part of the sternum, and the thymocytes or the nave thymic cells usually arises in the third and the fourth pharyngeal pouch, and here the yolk sac stem cell which contains the precursors of thymus or the T lymphocytes usually move on to the fetal liver and then to the bone marrow and ultimately to the thymus, where these T cells usually get mature to form the thymocytes. So they are called as thymic lymphoid cells or the thymocyte. In the first organ that is said to be lymphoid in nature. In the human, the maximum size of the thymus will reach just before the birth, and it keeps on increasing at the age of 12, and after puberty there is a successive progressive involution of the thymus. That indicates that the antigenic challenge that is given to the thymus to mount a heightened immune response takes place only in the early part of the life, and it is after the puberty there is a successive progressive involution where you can see there is a decrease in the cell mediated type of immunity, and this is usually present behind the upper part of the sternum. And then coming to the function of this thymus, it is the one that usually produces the T lymphocyte that usually carry out the cell mediated immunity, and they are called as the thymic lymphocytes, and it is a place where the thymus usually makes the T cell to proliferate and differentiate and add certain markers on the T lymphocyte to go and interact with specific type of antigen. So the antigen that is added is called as the THY antigen. So it is the first antigen that can be seen on the T lymphocyte that can go and interact with the foreign particle, and these cells are usually called as T cells because they are usually thymus dependent, that is they are generated in the thymus, that's why they are called as T lymphocytes or the T cells. And the immunocompetence, that is the thymocytes are usually naive, that is they are not functional in nature, and they get maturity only after the antigenic stimulation. So it is after the antigenic stimulation that the T cells become immunocompetent to mount an immune response. So before, it is a pre-thymic cell that are educated in the thymus to develop the markers against the foreign antigen, but not towards the self antigen. So these cells that are able to mount an immune response are called as immunocompetent cells. And the important function of the thymus can be known when there is a thymectomy, that is a removal of the thymus. And when there is a removal of the thymus, you can see a drastic decrease in the cell mediated immunity, leading to different types of disorders, you can say, which are called as lymphopenia, or it can be like congenital aplasia that usually says about the decrease in the cell mediated immunity. And in humans, there is a thymectomy or loss of thymus function leads to Dye-Georges syndrome, and in the mice it is called as a nude mice. And these thymic cells or the thymus derived T cells are mostly seated in the secondary lymphoid organ in particular areas. So wherever they are usually deposited, it is called as thymus dependent area, and where you can see the B lymphocyte in more number, it is called as a T independent area. So the T dependent area in the peripheral lymphoid organs are usually the white pulp, or you can see them in the central arterioles or the paracortical region of the lymph node. So they are called as thymus dependent area. And the thymectomy is the one where there is a removal of the thymus to know about the importance of the thymus participating in cell mediated immune response. So there is a decrease in the cell mediated immune response. Along with the decrease in the cell mediated immune response, you can also see that the other lymphocyte also being influenced due to the decrease in the cell mediated immune response, because there is an interaction of different cells, and it is the T lymphocytes that usually secrete certain types of lymphokines that activates the other lymphocytes too. So once there is a decrease in the cell mediated immunity, then definitely it usually affects the other lymphocytes too. And during the selection process in the thymus, only 1% of the thymus or the thymocytes or the T cells usually come out successfully, whereas the other T cells are usually eliminated by a process which is called as wasteful process. So these T cells that are usually eliminated are the ones that may have certain antigen that reacts with the self antigen, or they may have certain types of antigen that may bring about a cross reaction, or they may have a antigen that is not of any useful purpose. So these T lymphocytes have to be eliminated, otherwise the deposition of these cells can cause a damage to the human body. So these are usually eliminated by a selective process, and it is called as a wasteful process, and only 1% of the T lymphocytes or the T cells usually come out successfully that are able to mount an immune response. And it was Gill who proposed us the concept that the thymus plays a vital role in the development of cell mediated immunity. So thymus is a primary organ that helps to develop the lymphocytes, that helps in proliferation, differentiation to make it immunocompetent to give an heightened immune response against the pathogenic group of organism. And the cells that are developing from the thymus are called as the T lymphocytes. Now coming to next important central or the primary lymphoid organ is the bursa fabriceus. So it was first discovered in the birds, and it is a lymphoepithelial organ that arises as a pouch on the dorsal part of the cloaca. And here in this case of bursa fabriceus, because it was first found in the birds to give a humoral immune response, the cells that are usually developed from this bursa fabriceus are said to be B lymphocyte. So similar type of function is seen in the humans in the bone marrow, and hence the lymphocyte that arises from the bone marrow are called as the B lymphocyte. And the difference between the B lymphocytes and the T lymphocyte is the development and maturation of these lymphocytes in a specific organ. That is, in case of T lymphocyte, because they develop and mature in the thymus, they are called as T lymphocyte. And in case of B lymphocyte, because they develop in a bursa fabriceus in birds and bone marrow in the human being, they are called as the B lymphocyte. Now coming to the generation, how these B lymphocyte usually arises. So it is a stem cell that is a precursor of the B lymphocyte moves onto the yolk sac in the early gestation, and then move to the fetal liver and into the bone marrow, and ultimately reaching the bursa, where the B lymphocytes usually proliferate, differentiate, and become immunocompetent to finally form the B lymphocyte, which expresses a marker which is called as immunoglobulin. Now this depending upon the type of antigenic challenge, that type of B cells will be produced to produce a plasma cell, and these plasma cells are the one that produces the antibodies which goes and interacts with the antigen. So these cells which are able to mount an immune response are called as immunocompetent B cells. So before that they are called as naive cells, but once they interact with the antigen and produce specific type of antibodies, they are called as immunocompetent B cells. These bursa fabriceus derived B lymphocyte are also seeded in a particular areas in the peripheral lymphoid organ where they mount an immune response. So they are selectively seeded in the thymus independent area, mostly in the mantle, you can see in the germinal follicles, and in the peri-follicular region where the B lymphocytes are rich and highly dense in nature. So this is called as B dependent area and also called as thymus independent area. Now the B lymphocyte once it interacts with the antigen, it differentiates into plasma cell, which is the effector cells of the humoral immune response, and these plasma cells are the one that produces the antibodies. And the function of the bursa is to carry out the humoral immunity. Because antibodies are mostly seen in the body fluids, the function is said to be carried out of a response which is called as the humoral response, and the immunity is called as the humoral immunity. Similar type of function is seen in the human body carrying out in the bone marrow, and hence the lymphocyte derived in the bone marrow matured, that is the produced and matured in the bone marrow, are called as the B lymphocytes. So here the B lymphocytes are generated in the bone marrow, and it is in the Peyer's patch that develops that you can see the antibodies developing in the Peyer's patch, later seen in the secondary lymphoid organ, that is in the spleen and the lymph node, where they usually interact with the antigen to mount an immune response. So in human fetus, first the lymphocyte develops in the Peyer's patch and then move on to the peripheral lymphoid organs to mount an immune response. So the basic central or the primary lymphoid organs that develops the lymphocytes are the thymus and the bursa fabriceus in the birds, and it is the bone marrow in the human being. So both of these lymphoid organs, that is the primary lymphoid organs, helps in the development of lymphocytes. Now these are the areas where the lymphocytes are generated, they are mature to become immunocompetent. When they have become immunocompetent, they move to the peripheral lymphoid organ via the blood and the body fluids to reach there to mount an immune response towards the antigenic challenge. So the function of the secondary lymphoid organs is to present the lymphocyte with the antigen. So wherever the antigen is entering into the body, it is the secondary lymphoid organ where the antigen is captured, and it provides a ground where the lymphocytes can interact with the antigen to eliminate the antigen or to bring about the immune response. So here we have the secondary lymphoid organs. So here the secondary lymphoid organs can also be classified as most organized and scattered ones. So the organized ones are usually the Peyer's patch, and we have the scattered one is mostly the mucosal associated lymphatic tissue. So among the organized, we can also study about the spleen and the lymph nodes. So here the lymphatic, the major secondary organ that mounts an immune response towards the antigen and provides a ground for the interaction of antigen with the lymphocytes are the lymph nodes. So lymph nodes are nothing but the nodes that are usually formed at the junction of the lymphatic vessels. And whenever the antigen enters into the body, the antigen is captured in the lymphatic vessels and brought to the lymph node, where the interaction can take place between the lymphocytes that are now immunocompetent to mount an immune response. So the structure of the lymph node is that it has lobes, two lobes, which are usually encapsulated with the fibrous capsule interior to which develops a septa, and this septa divides the lymph nodes into two major components, the outer cortex and the inner medulla. And here the outer cortex usually consists of majority of the small lymphocytes which are still proliferating and differentiating, whereas the inner medulla usually consists of the mature lymphocytes. So here in the medullary cords and the follicles that is in the outer cortex, follicles, the primary follicle usually consists of the B lymphocyte. Now this primary follicle has the ability to develop into secondary follicle along with the germinal center. It is the germinal center where the clonal selection of the cells usually takes place. That is, the cells which are able to mount an immune response are only allowed to come out, whereas the others are usually destroyed. So in the follicular region, that is the primary follicle, and in the medulla, you can see they are rich in B lymphocytes. That's why they are called as B dependent area. So between these two areas, there is a paracortical region which is rich in T dependent lymphocytes or the T lymphocytes. So this area is called as a T dependent area. So whenever there is an antigenic challenge, the antigens are usually brought to the lymph nodes in different parts of the body, and these lymph nodes are widely distributed throughout the whole body, and they help to mount an immune response in different parts of the body. That is, you can say they are the one that mounts a systemic immune response towards a particular antigen. So wherever the antigen enters, they are brought to the nodes, and it is a place which helps in interaction with the antibodies to eliminate the antigen and mount an immune response. Now next, coming to another important secondary lymphoid organ is the spleen. Spleen is the one which is the largest lymphoid organ, and it is the one that usually clears the organism which are the blood-borne antigen. So whenever the antigen enters the blood is directly taken to the spleen, and the spleen is the one that usually filters the antigen from the blood. So it is able to remove the blood-borne antigen. Similarly, lymphatic system or the lymph nodes are the one that filters the antigen from the lymph. So here in case of spleen, they are the one that eliminates the antigen from the blood. So blood-borne antigens are eliminated, and the spleen is again most organized secondary lymphoid organ, consisting of the capsule, and here you have two major components that are divided as red pulp and the white pulp, and the capsule usually interior to the capsule there is a projection of tubercula which divides the spleen into two major components. One is called as the red pulp and the other one is called as the white pulp. The name red pulp is usually given because it is a place where the dead RBCs are eliminated, and it is a graveyard for RBCs, whereas the white pulp is the one which contains the splenic artery having a periarterial lymphoid sheath, and it is the lymphoid sheath which contains the primary follicle and the germinal center and also the B dependent area. Now whenever the antigen enters, they are directly through the blood, they are directly taken to the spleen, and it is in the periarterial lymphoid sheath where you can see the interaction of the lymphocytes with the antigen, and it is a graveyard for the blood cells, and it is the one that usually brings about the systemic filter. It acts as a systemic filter to remove the blood-borne pathogen. And between the red pulp and the white pulp, there is a marginal zone which is usually highly dependent on the T lymphocyte. So here we have T lymphocyte along with the interdigitating dendritic cell that presents the antigen to the T cell to mount an immune response. So spleen is next organized like secondary lymphoid organ to eliminate the antigen from the blood-borne antigens. Next is the mucosal associated lymphoid tissue, and here again the mucosal associated lymphoid tissue can be somewhat organized one, the other one can be a little bit scattered one. So somewhat organized one is the Bayer's patch, and the mucosa is a layer that usually covers the respiratory tract or the gastrointestinal tract, the genitourinary tract. So it is more prone to the different types of antigens. So there is an antigenic challenge that is at heightened state, you can say in the mucosal associated lymphoid tissue. So here we have a specialized mucosal associated lymphoid tissue which are the Bayer's patch, and the mucosal associated lymphoid tissue such as in the livers you have lymph node, you have tonsils, etc. So these are the region which interacts mostly with the antigen and produces immune response towards that particular antigen. Apart from that, you also have gut associated lymphoid tissue mostly consisting of the adenoids and the tonsils, and the follicles of the colon, and then we in this particular region we have a combination of cells which are both lymphocyte as well as the phagocytic cells. And in case of this B lymphocytes that usually produces the antibodies, the most prominent antibodies that are seen in the mucosal associated lymphoid tissue are the IgA antibodies, which are secretory in nature, and these are the only ones that are able to cross the mucous layer to provide immunity against different types of antigens. So IgA is a prominent antibody that can be seen in mucosal associated lymphoid tissue. And when an antigenic challenge takes place in this mucosal associated lymphoid tissue, because it is a connected lymphoid tissue, the antibody produced at one area also induces the production of antibodies at the other area. So it gives us a superiority of oral or nasal immunization. So when an oral or nasal immunization is usually given, it is seen to it that the whole body responds towards that particular antigen and mounts a generalized immune response in the human body. So mostly in case of vaccinations, you can see there is a oral and nasal immunization towards a particular antigen, and the whole body develops immunization against a particular antigen. So these are all about the organs of the immune system. So in the organs of the immune system, we have studied about the central and the peripheral lymphoid organs. The central comprising of bone marrow, thymus and the bursa fabricis, whereas the secondary lymphoid organs, it is mostly the lymph nodes, spleen and mucosal associated lymphoid tissue. Apart from that, you can also see cutaneous associated lymphoid tissue, that is related to the dermis, that is the skin. So whenever there is an antigenic challenge on the skin, the cutaneous associated lymphoid tissue is the one that captures the antigen and brings it to the node to mount an immune response. So all these are nothing but the organs of the immune system that helps in the generation of the lymphocyte, maturation of the lymphocyte to mount an immune response by making them immunocompetent. Immunocompetent are the cells which are ready to mount an immune response towards that particular antigen. So without this particular organs, there is no development of lymphocyte and there is no immune response or immunity in a person. So next we'll be studying about the next important component of the immune system, that are nothing but the cells of the immune system. So as already discussed, the cells are distinct and diverse in nature that carries out different function in the immune system, and they are widely divided as lymphoid cells and reticuloendothelial cells. Lymphoid cells are the one that comprises of lymphocytes and the plasma cell that mounts a specific immune response, whereas the reticular endothelial cells are nothing but the phagocytic cells that belongs to non-specific immune system that helps to eliminate the antigen by a process called as phagocytosis. And apart from acting as a non-specific immune component, it also helps to activate the specific immune response. So this is a system, you can say, that straddles between both innate as well as acquired immunity. So coming to the cells of the immune system, the most prominent cells are the lymphocytes. Lymphocytes are small and they are round in nature, of about 20 to 25% of the total lymphatic system in the lymphatic, that is in the lymph. You can see them about 20 to 25%, and they are about 10 to 12 types of lymphocytes that can be seen in the lymph, and they are usually divided based upon their sizes as small lymphocyte and large lymphocyte. And the major function of the lymphocyte is to mount an immune response towards the antigen. So lymphocyte are of various type, having different markers to go and interact with a specific antigen. So before going on in detail about how they interact, let us see the characteristics of these lymphocytes. So based upon the size, you can classify them as small lymphocyte and large lymphocyte. Small lymphocyte about 10 to 12 micrometers, whereas the large lymphocyte about 15 to 20 micrometers. And these small lymphocytes are usually spherical nucleus, and they show slow motility, and they have the least complex structure when compared to the large lymphocytes. So large lymphocytes are the one that can participate in immune response, whereas the small lymphocytes are the one that are least in the structural complexity when compared to that of the large lymphocyte. And depending upon their age, how long do they survive in the immune system, they can be classified as short-lived lymphocyte and long-lived lymphocyte. Short-lived usually survives only for few days, whereas long-lived can be present in the body for months together and also till the person lives, that is till he dies, these lymphocytes can be present in the human body. So short-lived lymphocytes are mostly nothing but the effector cells that usually are present whenever there is an antigenic challenge. Once the antigenic challenge subsides, the small lymphocytes are eliminated, whereas the long lymphocytes are the one that is mostly the, you can say, the memory, immunological memory cells that can keep circulating in the body to provide us protection against the subsequent infection. So mostly the long-lived lymphocytes are the T cells. And coming to the lymphopoiesis, lymphopoiesis is nothing but the generation of the lymphocyte, as already seen in the structure of the immune system, that is the lymphoid organs. These lymphocyte generates in the lymphoid organs as a yolk sac stem cell, and they move to fetal liver and then to the bone marrow and to the thymus if they are T derived, or they move to the bone marrow only to be B derived, that is the B lymphocyte or the T lymphocyte. So the lymphopoiesis is the generation of the lymphocyte, and after generation they are made immunocompetent to mount an immune response. So these lymphocytes keeps on circulating in the body fluids such as blood and the lymph, and they act as a policeman which are on a beat patrol to keep circulating in the body for about certain period of time to see or check whether there is any type of infection in the body. And whenever there is an antigenic challenge, these lymphocyte usually goes to that particular area and they mount an immune response towards that particular antigen. And these lymphocytes usually rotate or circulate in the body in a phenomena which is called as lymphocyte recirculation. Lymphocyte recirculation means that there are different types of lymphocyte carrying out different types of function, but they do not move separately or distinctly. They mix together in a population or as a mixed population and keeps on circulating in the body fluids such as blood and the lymph to check or to ensure that if there is any antigenic challenge, they move to that particular area and interact with the specific antigen depending upon the type of lymphocyte. So here we have the immune response that is usually mounted against the antigen and it is a lymphocyte recirculation that ensure that there is an immune response wherever there is an antigenic challenge. And this recirculation takes place only of about 1 or 2 days, and it is the T cells or the B cells that usually brings about the recirculation or the lymphocyte recirculation. Apart from that, these cells expresses certain markers on their surfaces, that is a lymphocyte expressing certain markers on the surfaces to become immunocompetent, that is they are able to mount an immune response at a specific antigen. And it is a hallmark of adaptive immune system that towards a specific antigen only that type of lymphocyte can go and interact. So it is a surface markers that are usually present on the lymphocyte that helps to recognize the antigen and interact the antigen in a specific manner. So these lymphocytes can be broadly classified into two types, that is one is called as a naive lymphocyte and the other one is called as a mature lymphocytes. So before the lymphocyte interacts with the antigen, it is called as a naive lymphocyte, that is it is not immunocompetent in nature, that though it does not go and interact with the antigen, it has certain properties, that is they helps in recognizing the antigen, they brings about immunological memory and helps in mounting an immune response. So once these lymphocytes, that is a naive lymphocyte, interacts with the antigen, they become mature lymphocytes. And these mature lymphocytes can be discriminated or distinguished from the naive lymphocyte by the expression of the markers on their surfaces. So these are called as the surface markers, and based on these markers you can recognize the antigen and the antigenic specificity of that particular lymphocyte with that particular antigen. So when there is an interaction between the immunocompetent cell and the specific antigen, it can either lead to tolerance or it can lead to an immune response. So tolerance usually comes when the antigen is in very small amount, and also when the antigen is a self antigen, because the lymphocyte have been educated in the primary lymphoid organ to not to produce the activity against the self antigens. That is what is called as immunological tolerance. Tolerance can be brought about either when there is a low amount of antigen or when the antigen is a self antigen. Now towards the non-self antigen, when the immunocompetent cell interacts with the specific antigen, the immune response is developed only towards the non-self antigen. The immune response, as I told you, can be either a cell-mediated response or a humoral response. So here in the cell-mediated response, it is usually brought about by the T cells, and in the humoral response, it is brought about by the B cells. So when there is B cell immunity or the humoral immunity, the effector cells that are produced from the B cells are called as the plasma cells, and these plasma cell produces the specific type of antibody that goes and interacts with the antigen. Now coming to the T cells, when the antigen interacts with the T cells, again it helps in the production of the effector cells, and the effector cells of the T cells are of two types. It can be a Th cell or a Tc cell, and the Th cell is the one that is the important cell in the immune response that produces the lymphokine or the cytokine, helps in the generation of the other lymphocytes. So based upon the nature of the immune response that is developed, the immune response can be classified as T response or the B response, and it takes place only when there is an interaction between an immunocompetent cell and a specific antigen. Now the surface markers helps us to know the stage of differentiation and the functional property of that particular cell. So based upon this, you can classify the cells as T cells and the B cell. First is the classification based upon where they usually originate, whether they are in the thymus or the bone marrow, and the differentiation, how do you distinguish these cells from one another, is by the surface markers that are present on the T cells and the B cells. So the surface markers that are expressed are usually were given different names initially, but later on a specific nomenclature system was given to name these markers as CD cells, that is cluster of differentiation cells. So these cluster of differentiation cells are nothing but they are usually identified as marker by using monoclonal antibody. So cluster of monoclonal antibody, when it goes and interacts with a specific antigen, you give that particular term, that is called as CD, cluster of differentiation. So there are about 150 CD cluster of differentiation that can be seen on different types of lymphocyte. So based upon this CD, you can classify the B cell markers and the T cell markers. So let us see the difference between the T cell markers and the B cell markers. So this is how you distinguish between the T cells and the B cells. So T cells usually have the CD2 receptor that is usually goes and binds with the sheep erythrocyte, whereas this CD2 receptor are absent in the B cell. Whereas B cell recognizes sheep erythrocyte when they are coated with the antibody, and the receptor that recognizes this coated antibody is a CR2 receptor, which is absent in the T cell. And the T cell expresses a specific receptor which is called as T cell receptor, TCR, whereas in the case of B cell receptor, it is the immunoglobulin that can be seen. Mostly it is the IgM or the IgD that is expressed in the early part of the life. So Ig is on the surface. Immunoglobulin receptors are nothing but they are receptors which are present on the surface and absent in the T cell, whereas T cell contains a T cell receptor which is called as a TCR, and a specific antigen can be seen on the T cell which is called as Thai antigen, whereas this type of antigen is absent in the B cell. And the blast transformation can take place in the T cell by using metogens such as concavalin A, etc., whereas the blast transformation of the B cell takes place with the help of bacterial endotoxins. And next is the T cells are free of any cytoplasmic projections, and the B cells usually have filamentous surface and microvilli that helps them to distinguish from the B cells and the T cells. Now next, coming to how these T cells usually mature, and these express surface markers on the surface to interact with the antigen. So here the T cell maturation usually takes place as already discussed. Stem cell, usually the prominent earliest T cells that are produced are CD7 plus cell, which moves to the thymus and develops a CD2 receptor. Now this becomes a pro T cell. Now from the pro T cell they move on to the cytoplasm to form the pre T cell expressing TCR, that is T cell receptors. Now these T cell receptors can discriminate into two different types. One is having it is a heterodimer, you can say, having alpha, beta or gamma and delta chain. So depending upon the chain, the T cells can be classified. So mostly seen T cells are the one which have a combination of alpha, beta rather than gamma and delta. Now this TCR having alpha, beta again differentiate into two different types. One is called as the TH and the TC cell, whereas the TC delta are the one that are mostly used in immunosurveillance, and much important functional property is known about the TCR, delta and gamma, whereas TCR, alpha, beta are mostly studied one, and they can be classified as a TH cell or the TC cell. So TCR is a heterodimer and it has CD3 antigen marker and made up of two glycoproteins, alpha, beta and gamma, delta, and the TCR has a 4 region of variable, diverse, joining and the constant domains, and VDJC is the one that rearranges themselves to produce a specific type of T cell that interacts with the diverse group of antigens. So rearrangement of these chains helps in developing receptor against a specific type of antigen. So because they contain VDJC chain, they are called as immunoglobulin gene superfamily, or they belong to immunoglobulin gene superfamily. And the reassortment is very essential to identify different types of antigen on a specific organism. So they are diverse group of antigen. So in order to identify that, you have got different types of T cells, and these T cells have rearrangement in the VDJC chain to produce the marker to interact with a specific antigen. So again, depending upon whether it is a self antigen or a non-self antigen, there is a development of self-tolerance and most of the T cells that interacts with the self antigens are eliminated, but still these forbidden clones are being produced and they are usually suppressed by some immunosuppressive cells. So it is when there is a breakdown in this regulation of producing the self lymphocyte against self antigen, there is leading to the autoimmune diseases. So there is a regulation or a check that takes care to see that self antigen lymphocytes are not produced to interact with the self antigen, and the most important concept of the elimination of the self antigen is in the thymus, or the process is called as the elimination or the wasteful procedure. Now based upon the MHC restriction, that is there are certain T cells that recognizes MHC molecule on the antigen presenting cell. MHC molecule is nothing but major histocompatibility complex that is present on the antigen presenting cell. So there are certain T cells which are of CD4 plus and CD8 plus. So T cell having CD4 plus cell recognize class 2 MHC molecule, and they are called as the TH cell, that is they are called as T helper cell or inducer cell. And the TC cell which have CD8 plus receptor can recognize class 1 MHC molecule on antigen presenting cell, and they are called as TC cell. Now the difference between the B cell and the T cell is that B cell can recognize the antigen directly, whereas the T cell cannot recognize the antigen directly. It requires the help of an antigen presenting cell that expresses similar type of the component that is acting as self antigen on the MHC molecule. So the T lymphocyte recognizes antigen when it is presented only with the MHC molecule. So MHC molecule is again class 1 and class 2, and the topic about the MHC will be dealing in later. But remember that T cell interacts with the antigen only when it is in combination with either class 1 or class 2 MHC molecule. So T cell having CD4 receptor can recognize antigen when they are presented along with class 2 MHC molecule, and T cell with the CD8 receptor recognizes antigen when they are with the class 1 MHC molecule. So based upon that, they are called as T helper cell or inducer cell and T cytotoxic cell. So the main function of the T helper cell or the inducer cell is to induce an immune response or to activate the other cells of the immune system by producing certain types of lymphokines or the cytokines. So the cytokines that are produced are of different types, and these cytokines helps in the activation of macrophage macrophages, helps in the activation of B cells. Along with that, they also helps in the activation of the TC cell, that is the cytotoxic T cells. Based upon the cytokines that are produced by the TH cell or the inducer T cells, you can classify the TH cells into TH1 and TH2. The TH1 cell produces the cytokines such as interferon gamma, interleukin 2 that activates the macrophages. Apart from that, they also helps to produce the cytokines that helps in the intracellular killing of the tubercle bacteria or the lepra bacilli. So next, TH2, the cytokines produced by the TH2 are interleukin 4, 5 and 6. They are the one that helps in the development of the other immune cells, that is they help in activating the proliferating B lymphocyte and the other cells of the immune system. Now next, coming to the other type of the T cells or the sub T cells, they are called as the suppressor T cells. So they are the one that carries the CD8 marker on the surface, recognizes class 1 MHC molecule, and the function of the suppressor T cell is to suppress the immune response or to regulate the immune response and to check the immune response. If there is a loss in the regulation of the immune response, it can lead to severe damage to the human immune system along with the autoimmune diseases. So the main purpose of the T suppressor cell is to check the immune system or regulate the immune system. Next are the cytotoxic T cells, which are called as the TC cells, and they are the one that are usually carrying CD8 markers, and again class 1 restriction, that is they recognizes antigen when they are presented only with the MHC class 1 molecules, and it is this cytotoxic T cells that are cytotoxic towards the tumor cells and are the one that are involved in the allograft rejection. That is, allograft rejection is nothing but the tissue or organ that is transplanted from one person to the other person. So when there is a rejection, it is because of this cytotoxic T cells. So involved in the allograft rejection, and apart from that, they are the one that eliminates the virus infected cells in the body. So mostly targeting the tumor cells or the malignant cells in our body and the virus infected cells. So apart from these subsets of T cell, we also have memory cells. So whenever there is an immune response, the immune response is by producing two important cells. One is the effector cell that immediately interacts with the antigen, eliminates the antigen, and the other subset of population are the memory cells that keep circulating in the body to face the antigenic challenge upon the subsequent infection. So these memory cells are of both CD4 and CD8 types, and they keep circulating to protect the body against the subsequent infections. Now next, coming to the B cell maturations, the B lymphocyte, the stem cell reaches the fetal liver in the early gestation, and later they get transferred to the bone marrow, where they usually develop to produce the B lymphocyte, and the pre-B lymphocyte produces IgM in the cytoplasm, and later part of the life it is expressed on the surface as an marker on the B cell. That is, IgM inside initially it is present in the cytoplasm, later on it is expressed on the B cell as a marker. So the first marker that is expressed on the B lymphocyte is the IgM. Later on there is a isotype switching, where apart from IgM you can also see the IgG type or IgG immunoglobulin on the B cell. So it is due to the rearrangement in the different chain, you can see the B cell producing different types of immunoglobulin markers against a specific antigen. So once the specificity is between the antigen and antibody is perfect, then the B cell produces the plasma cell to produce the antibody of that specific type towards that specific antigen. So apart from isotypic switching, you also have allelic exclusion, where as we have seen in the antibody molecule, there are two chains, the heavy chain and the light chain, and the light chain comprises only of either kappa particle or the lambda particle. So due to this allelic exclusion, there is either a kappa chain or the lambda chain that will be present on the immunoglobulin molecule or the antibody molecule. So again here, whenever there are self antibodies that are produced towards self antigen, there is elimination of these self reacting antibodies by a process which is called as a clonal selection or anergy. And whereas the B cell that interacts with the antigen or a specific antigen or a pathogenic antigen will survive and reacts with the antigen to produce the humoral response by producing the plasma cells, and these plasma cells are the one that produces the antibodies. The plasma cells are more complex in nature. They are more developed one. They are double the size of the normal small lymphocytes, and they have more structural and chemical complexity. They have well developed endoplasmic reticulum, Golgi complex. They have a very distinct nuclei which is present in the plasma cell. So the plasma cell can be discriminated with the name cell by the most structural complexity, and it is the plasma cell that produces the antibodies or the specific antibodies towards the specific antigen. The lifespan of the plasma cell is very short, so it is only during the antigenic stimulus where you can see the production of the plasma cell. Once the antigenic stimulus is over, once the immune response has taken place, the plasma cell disintegrate or degenerate, whereas the memory cells keeps on circulating in the body upon subsequent infections. So there is another lineage of a B cell that expresses a specific marker which is called as a T cell markers. They are the one that are involved in producing the autoantibodies, and one of these B cell which is called as a B1 cell that expresses CD5 on its surface is the one that is involved in autoimmune diseases. So B cell maturation, it is the one that synthesizes the B cell, expresses immunoglobulin on its surface, and produces a humoral response by producing the plasma cell, which produces the antibodies of specific type towards a specific antigen. Next, coming to the another important group of lymphocytes which are called as the null cells. So they are about 5 to 10% of these null cells in the lymph that keeps on circulating, and they are the one that belongs to non-specific immune response, and they are completely different from the B cells and T cells. They are produced by a different lineage, and they do not express or they carry out a specific immune response or a humoral or an cell-mediated immune response, but they have their own distinct property and function that is usually carried out in the human immune system. So null cells are also more the one which have a complex structural complexity, well developed components inside its cell along with the eosinophilic granules, and these are also called as the natural killer cells, and they are the one that targets against a variety of tumor cell and the virus infected cells in the body. So based upon their function, the null cells can be classified as the natural killer cell or antibody dependent cell mediated cytotoxic cells, or they can also be called as lymphokine activated killer cells. So they all of them have the similar function, and it is a natural killer cell that is mostly called as the null cell. So target cells of these natural killer cells are usually the malignant cells or the tumor cells that are developed inside the body, and the virus infected cells. So these natural killer cell eliminates these malignant cells and the virus infected cells, and they are said to be natural and they do not require any antigenic stimulus. So they are non-immune in nature, belonging to the innate immune system, and they are the one that are mostly involved in the severe combined immunodeficiency disease, which are called as the SCIDs disease, because they do not require any antigenic challenge, and they express two type of markers on its surface which are called as CD16 and CD56. So these are called as glycoprotein receptors, and they have a cytolytic factor which is called as perforin. So similar to that of the complement system, C9 complement system, which forms a hole on the surface. So these perforin usually forms a hole on the surface, and the tumor necrosis factor enters into the cell, and the tumor necrosis factor brings about the lysis of the tumor cell or the virus infected cell by a process which is called as apoptosis. Apoptosis is nothing but programmed cell death. So it is the death of the tumor cell or the virus infected cell which is in a programmed manner, so without involving any other cells in that vicinity. So it is by apoptosis that natural killer cells usually eliminate the tumor cell or the virus infected cell, and it is the perforins that are present in the eosinophilic granules that causes the pores on the cell, and tumor necrosis factors brings about the lysis. So usually these natural killer cells are said to be the one acting against the virus infected cell because they are activated by the interferons. Interferons are nothing but the natural substances that are produced by the human body in response towards the viral infection. So whenever there is a production of the interferon, indicates there is a viral infection. So immediately the natural killer cell gets activated to remove the virus infected cells. And apart from that, they are also involved in the immune surveillance to eliminate the virus infected cells or the tumor cells. Next group of the natural killer cells are the antibody dependent cell mediated cytotoxic cells. So these are nothing but these cells have receptor on the surface which are complementary to the FC portion of the antibody. So when there is an antigen antibody complex, the receptor, that is FC portion of the antibody molecule, like it goes and interacts with the ADCC molecule and helps in enhancing the phagocytosis. So that is called as antibody dependent cell mediated cytotoxicity. So antibody is required for the cytotoxic of the antigen to take place. So it is called as the antibody dependent cell mediated cytotoxicity. Apart from that, the null cells are also called as lymphokine activating killer cells. They produces interleukin 2, and this interleukin 2 helps in identifying the tumor cell and eliminating the tumor cell only without affecting any other cells in the vicinity. Now these lymphokine activating killer cells are mostly used to treat different types of tumors inside the body. And next we have phagocytic cells, the most important group of cells that belongs to both innate as well as specific immune response. So phagocytic cells are nothing but mononuclear macrophages that keep circulating in the body in the blood as monocytes, and when they reach the specific tissues, they usually forms histiocytes. So macrophages can be either in the blood as monocytes, or they can place themselves in specific tissues in different parts of the body as macrophages. So they are called as blood macrophages. Monocytes usually are of about 12 to 15 micrometers, and the tissue macrophages are larger when compared to the blood macrophages, and they are about 15 to 20 micrometers. And they are usually produced by the bone marrow monocytes, and when they generate in the bone marrow as monocytes, they circulate in the body fluids for about 6 days. Now once they keep on moving, they have a half-life of only about 3 to 4 days. So before 3 to 4 days, they go to a specific tissue in the body and they establish themselves in that particular tissue. Take for example the alveolar macrophages that usually resides in the lungs to protect against the tubercle bacteria. And apart from that, the macrophages that are present in the kidney are called as the Kupffer cells. So depending upon the different places where they establish themselves, they carry out different function, they are given that particular name. So monocytes are the one that circulates in the body, in the blood, whereas the histiocytes or the tissue specific macrophages are the one that have already established in a particular tissue and carrying out certain functions. And these monocytes or the macrophages usually express certain types of markers on them. So these markers are called as IA proteins, the FC proteins, or the FC receptors that have specificity towards the FC portion of the antibody, and they also have receptor for certain complement products and also receptors for certain types of lymphokines. And the main function of the phagocytic cell is to carry out the phagocytosis. So phagocytosis is nothing but the engulfment of the microorganism and degrading the microorganism into variable lengths of fragments. So the initial step of engulfing the microorganism is to reach the microorganism in that area wherever there is an antigenic stimulus. So this is brought about by a procedure which is called as the chemotaxis. The phagocytic cell move towards the organism by a process which is called as a chemotaxis. Once it interacts with the antigen, it extends its pseudopodia to enclose the organism in a membrane or a vesicle which is called as a phagosome. Now interior to this phagocytic cell, the phagosome fuses with another important vesicle which is called as a lysosome. Now the fusion between the phagosome and lysosome produces a larger vesicle which is called as phagolysosome. Now the lysosome contains lytic components that lyses the bacteria or any organism into fragments, and these fragments are eliminated from the phagocytic cell which are by a process which is called as exocytosis. Now during this exocytosis, these phagocytic cells usually act as antigen presenting cell, where there are certain markers on the phagocytic cell which are called as MHC molecule. Either it can be a class 1 MHC molecule or a class 2 MHC molecule. So depending upon the expression of antigen on the phagocytic cell, it can either activate a TH cell or a TC cell. So the main purpose of the phagocytic cell is to phagocytize the organism, degrade the organism into variable fragments and eliminate the organism, making it inactive in nature. So because of the markers, they are able to activate other cells of the immune system, and the induction execution of the immune response is usually brought about by the macrophages, and it is the one that helps to process and present the antigen as with the help of class 1, class 2 MHC molecule, and the MHC genes which are present on the phagocytic cells are similar to the receptors or the TCR receptor that are expressed on the T cell. So whenever there is a compatibility, only then it is able to present the antigen to the T cell, whether it is a TH cell or a TC cell. And there is a difference between the inactivated macrophages and the activated macrophages. The inactivated macrophages are the ones that are usually normal in nature. Whenever there is no immunogenic reaction or whenever there is no antigenic challenge, they are usually in a normal form. Whereas when there is an antigenic challenge by different types of organism, the normal macrophages gets activated to form the activated macrophages, which are more potent in nature to phagocytize the organism more potently and eliminate the organism. So they are different, there is a change from the normal macrophages to the activated macrophage. There is an increase in the size, there is increase in the production of phagocytic enzymes, there is increase in production of different types of leukins that helps to activate the other cells of the immune response. And apart from this activation by the activated macrophages to eliminate the antigen, when the antibody is involved to activate the macrophage becomes armed, and these armed macrophages are mostly act as opsonin to enhance the phagocytosis or to bring about the phagocytosis of the particular group of organisms. So these are macrophages, and the other groups related to these macrophages are called as microphages. So the microphages are nothing but polymorphonuclear leukocyte, PMLs. So the examples of these PMLs are the neutrophils, the eosinophils and the basophils. Neutrophils again, they are actively phagocytic in nature and mostly involved in acute inflammatory response, and they are non-specific in nature. They can interact with any type of organism, not towards a specific group of organism. Next, coming to eosinophilic cells are eosinophils, mostly involved in allergic reactions and inflammatory response, and they have two types of granules that releases the content to bring about the pathogenic effect on the microorganism. Mostly these eosinophilic granules or eosinophils are produced during parasitic infection as well as during an allergic response, and the amount of these eosinophils that are present is less when compared to that of the neutrophils. It is the neutrophils that are usually produced in response towards different types of infection, and there is a constant increase in the number of the neutrophils, which is called as neutrophilia, indicates there is certain infection in the body, and because it is non-specific, we will not know the which type of infection it is, but later on with the help of different diagnostic procedure we can identify the pathogenic group of organisms. So just to know whether there is any infection or not, you can see the number of increased neutrophils in the body. And last one is the basophils, are the one that are involved in hypersensitivity reactions, and usually these basophil contains granules containing pharmacological active agents. So these basophil has specificity towards the mast cells. When they go and interact with the mast cell, they causes the degranulation of the mast cell to release pharmacological active agents such as heparin, anaphylactic agents that produces anaphylactic or atopic allergy in atopic individuals. So next, coming to the last type of cells that are the dendritic cells. So the dendritic cells are similar to that of the macrophages, but they are non-phagocytic in nature. They act as antigen presenting cell that process the antigen and presents it to the T cells, that is either the TH cell or the TC cell, but they are non-phagocytic in nature. And mostly they express class 2 MHC molecule, that means they activate the TH cells of the T-inducer cells, and they are pleomorphic in nature, and these usually have a germinal center where you can see the elimination of the lymphocyte lymphocyte which are of not important to the human body. So germinal center usually brings about the clonal selection and helps to select only the type of lymphocyte that are interacting with the antigen, and they are the one that present antigen to the T cells, and the B cells sometimes can also act as antigen presenting cell, mostly during the secondary immune response. Apart from that, you also have these dendritic cells which are called as Langerhans cells that are related to the dermis or the skin. So whenever there is an infection from the skin, it is a Langerhans cell that carries the antigen to the lymph nodes, which can mount an immune response towards this antigen that are entering via the dermis. So these are the dendritic cells. The next thing that we'll be dealing in about the structure and the cells of the immune system is the major histocompatibility complex. And the main function of the immune response is to eliminate, first is to recognize the antigen and eliminate the antigen. So now when coming to the tissue graft or transplantation of the tissue or the organ from one individual to the other individual, there is an allograft rejection. There is a rejection of the tissue or organ takes place. So this rejection is called as the allograft rejection, which was first studied in inbred mice by the Gorer, who found that there was a rejection of the tissue or the organ when it was transplanted from one strain to the other strain of a similar species, that is the same species. So this, like when he studied that in detail, he found that there are two blood group antigen. The blood group 1 antigen that is common in all the strain, and the other group antigen that is present in only few strains of a particular species. And this is this second antigen that is usually involved in allograft rejection and mounting an immune response towards the tissue or the organ that is being transplanted. When this was further studies were carried out by Dorset, he found that the leukocyte contains this human leukocyte antigen that is mostly responsible for the tissue graft rejection. And ultimately Benacerraf was the one who said that it is a gene that is the genetic basis for towards the response of a particular pathogen or the tissue or the organ is usually brought about by a gene which is called as immune response gene, IR gene. So for their contribution to the field of like giving us a concept of major histocompatibility complex, the three were awarded a Nobel degree. So now we come to know that between the similar strains in a particular species, there is a rejection of the tissue or the organ that is being transplanted. So it is mostly because of the human leukocyte antigen or receptor that is usually expressed on the human leukocyte. So these are the one that mounts an immune response towards the tissue or the organ that is being transplanted. So now when the genetic basis was studied, it was found that these human leukocyte antigens are usually present on a chromosome pair, and they have three important regions which is coding for three important types of genes producing different types of products. So class 1 gene, class 2 gene and the class 3 gene. So class 1 proteins are the one that are mostly involved in the acceptance or the rejection of the allograft. Class 2 are the one that is involved in the immune response, like which type of response has to be developed. It is usually present on that particular chromosome of class 2. And then we have class 3 that is mostly activating the complement system and the tumor necrosis factor, the heat shock proteins, etc. So here the major histocompatibility complex, the name histocompatibility complex is usually given because it was studied in the transplantation process. So it is the H2 antigen that was initially discovered. Later on it was human leukocyte antigen in the human body. And the major histocompatibility complex is nothing but cluster of compatible genes that are closely related. They are multi-allelic genes that usually determines whether which type of protein has to be synthesized in a particular immune response. So the HLA complex is usually present in the short arm of chromosome 6, and these HLA molecules are of three types: class 1, class 2 and class 3. So class 1 MHC molecule that are usually expressed on the antigen presenting cell are of HLA molecule class 1. So these class 1 molecules are two type of glycoprotein molecules made up of alpha and beta, which are non-covalently, that is alpha, that is heavy peptide chain, non-covalently linked to the beta beta 2 microglobulin. So there is a non-covalent interaction or non-covalent linkage between alpha and the beta. So the heavy peptide is made up of three globides, alpha 1, alpha 2, alpha 3, along with the C transmembrane protein, which usually traverse the cell membrane. And there is a group that is formed by folding of alpha 1 and alpha 2, and in this group it is the place where the antigen is usually presented to the T cell, whether it is a cytotoxic cells or the usually the T helper cell. So class 1 molecule, as already discussed, they are the one that presents the antigen to the TC cell, that is cytotoxic T cell, and it is the cytotoxic T cell that is mostly involved in the allograft rejections. Now coming to the HLA molecule 2, it is made up of heterodimer having alpha and beta, alpha 1, beta 1, alpha 2, beta 2. The proximal region is usually constant in amino acid, whereas the distal one is the one which is a highly variable amino acid, and again it forms a group where the antigen is presented to the TH cell, and the TH cell is the one that induces an immune response and act as a helper or the individual cells to produce the immune response towards different types of pathogenic group of organism. Class 3, as already discussed, they are the one that are heterogeneous in nature, producing different types of product to activate a complement system, heat shock proteins and tumor necrosis factors. So all of them involved in the immune system to mount a heightened immune response to eliminate the pathogenic group of organisms. So for the tissue graft rejection and about the major histocompatibility complex, it is mostly the class 1 molecules that are involved in the allograft rejection, and MHC molecule is nothing but major histocompatibility complex that determines which type of immune response has to be mounted towards different types of the pathogenic group of microorganism. And recently these HLA typing is mostly used to identify the paternity test or to know whether transplantation can be done between compatible people or not, and it is the one that helps us to know in which individual the transplantation will be successful. Apart from that, it is also used in anthropological studies, etc. So before it was blood group, blood antigen or the blood grouping that was mostly used to identify the different things, but now it is the major histocompatibility antigen or the MHC antigen, or it is the HLA antigen typing that is mostly used to identify based upon the genetic basis. So this is all about the cells of the immune system and the major histocompatibility complex.